Simple designs test one thing at a time, but biology is rarely one thing at a time. What if fertilizer only works when water is high? What if your treatment effect depends on the batch? Part 2 is about designs that handle complexity. 2 questions: does fertilizer help? And does extra water help? You could run 2 separate experiments, or you could test both simultaneously in a factorial design. 4 groups: fertilizer plus high water, fertilizer plus low water, no fertilizer plus high water, no fertilizer plus low water. Magic: you can detect interactions. Fertilizer only boosts growth when water is high. With one factor at a time, you'd miss this completely. In biology, factorial designs test drug combinations, gene knockdowns with treatments and dose response across cell lines. Before you buy seeds, answer this: how many plots do I need to detect a 20% growth difference with 80% probability at alpha equals 0.05? That's a power analysis. The inputs: expected effect size, acceptable false positive rate, desired power. The output: minimum sample size per group. Run this before the experiment, not after. An underpowered experiment is a waste of time. You likely miss the effect even if it's real. In clinical trials, power analysis determines enrollment targets. In genomics, it determines sequencing depth. You measure one tomato's height 3 times with a ruler. That's 3 technical replicates. They measure instrument precision, not biological variation. You measure 3 different tomatoes. That's 3 biological replicates. They capture the variation that matters. Only biological replicates increase your sample size. In RNA-seq, running the same library 3 times gives technical replicates. Running 3 independent biological samples gives biological replicates. Journals and reviewers care about the biological N, not the technical N. You plant treated plots on Monday and control plots on Friday. Monday was sunny, Friday was rainy. Now your treatment effect is confounded with the weather. The batch is the treatment. You can't separate them. In genomics, library prep batches, sequencing lanes and processing dates all introduce batch effects. The fix: randomize samples across batches. Or, if you can't, include batch as a covariate in your model. Combat and limmas remove a batch effect exist because this problem is ubiquitous. The experimental design checklist: sample size from power analysis, randomization to break confounders, blocking unknown nuisance variables, controls for baseline and validation, factorial layer for interactions, biological replicates for the real N, batch randomization to avoid confounding. Plan the garden before you plant. Every statistical method downstream assumes you did this right.